Okay, we got to get through this really quick, guys, so we can move into the lab. Okay, so number one, we are looking at the cell wall. What does the cell wall do? It keeps the shape of the cell, or acts as a frame for the cell. Okay, so supports the cell. Something like that, okay, about shape, support, or frame is good for the function. Okay, structure number three, okay, is the rough ER. They don't have to have written endoplasmic reticulum. If they did, wonderful. Okay, ER is good enough. Each one is worth 2 marks, one for the name and one for the function. Okay, so rough ER, and its function is to transport protein. Okay, number 4 is the smooth ER. Okay, and I will accept. Breaks down complex carbohydrates, detoxifies poisons, makes lipids, makes hormones, repairs membranes. Okay, I will accept any one of those functions as a function of the smooth ER. Synthesize, yeah, that makes lipids, yeah. Okay, questions on that one? All right, number 5 is the water vacuole. Okay, and its job, same as the cell wall, it supports the shape of the cell, uses turgor pressure to support the shape of the cell. Something about support and shape would be good. I'll say not for today, but in the future, yes. Yeah, that's fine. Yeah. All right, number 7 is the chloroplast. Carries out photosynthesis, makes food for the cell using photosynthesis. Photosynthesis has to be in there as a function of the chloroplast. Okay. Okay, number 10, the layer just inside the cell wall is the cell membrane. And I said several times in class, the words I'm looking for are controls endocytosis and exocytosis. They must have those words. That is the function of the cell membrane. Okay, they have to have those terms there. Endocytosis and exocytosis. Okay, number 11. This is the Golgi apparatus. Gets rid of waste. Anything about excreting waste, getting rid of waste, making vesicles for waste, anything about waste is good. No, I said you can't write that. Okay? I said gets rid of waste. I said garbage man, don't write that, but that's essentially what it does. Those are my exact words. It doesn't break them down, it only packages them. Okay, number 12 is the nucleolus. That one's got to be spelled right. Okay, nucleolus. Controls protein synthesis, makes ribosomes. I will accept either one of those. Combines and copies DNA. Yes, that's controlling protein synthesis. Yes. No, it doesn't copy RNA. It makes RNA by copying DNA. I know that seems kind of anal, but it's true. It doesn't synthesize the proteins. No, the ribosomes do that. No, that's what the nucleus does, not the nucleolus. Okay, any other ones? Today, okay. In the future, no. Okay. All right. So give them a mark out of 16. Okay. And then bring them up to this desk right here behind Blake and grab a Chromebook and sign in. All right, so what we're going to be doing is, same as before, same as the microscopy lab, we're going to be going to the bio lab, okay? There's 3 slides, actually 4. 3 slides that are important, 4 that people, the 4th one people just always ask, can we look at this? And I always say, okay, sure, okay? But it isn't going to be part of the lab. It's not going to be something that you have to do a diagram for, not something that's going to be part of any of the analysis questions, okay? So, the problem we're investigating has to do with the cell theory, specifically the second point of the cell theory, and that is that cells must carry out the basic functions of an organism in order to survive. Thus, they must have certain structures within their cells that carry out these basic functions. That's the problem we're investigating. You've got to put that in your own words in the form of a question, remember. Okay? That is a statement, not a question. Okay. Design, manipulated, responding, controlled variables. Okay, we are looking at today 3 different cells. Okay, they're coming from different organisms. Okay, because we're looking at a plant cell. We're looking at a generalized animal cell. And we're looking at one of your cells. Okay, so the first cell you're going to want to look at is the plant cell. Okay, and it is called monocot-dicot cross-section. Okay, that's what the name of the slide is called. Essentially what it is they've taken a leaf, okay, and they've sectioned it. So if this is my leaf, see, I'm not a good artist, okay? So the veins are through the leaf. That's just getting worse. Anyway, okay, what they've done is they've cut it like this, and they're looking at it on its narrowest dimension. So what you will see on the slide are 2 things that look like this. Okay? Those are 2 sections of a leaf, cross sections of a leaf. You want to look at the one that's the dicot, and I'll show you what that looks like here in just a minute so you know you're looking at the right one. Okay? Then you're going to be looking at a generalized animal cell. On the slide it says generalized animal cell, or it says stratified squamous epithelium, which is a really fancy name for that skin you see that's red and hurts like hell when you dust your knuckle, but doesn't really bleed, just stings like crazy. That's that stuff. It's just underneath your outer, okay, kind of epidermis. It's the layer underneath that, okay, that you can see when you dust a knuckle or scrape yourself really good. Okay, so that's what we'll be looking at. The cells in that are really, really small. I'm going to show you what it kind of looks like here, and then you'll know what you're looking for as well in the lab. Okay, the last one that you're going to do a diagram of, so you're going to do a diagram of this, you're going to do a diagram of the animal cell, and you're going to do a diagram of your own cheek cells. Okay, now that's as far as we can go with using your own cells. Okay, like when I was in high school, you'd prick your finger with a lance and look at your own blood. But there's, scarier things out there now than there were when I was in high school. Okay, we have to worry about like hepatitis and HIV and all kinds of other things. So we can't look at our own blood or anything like that. But what we can do is what they do in CSI, which is take a cotton swab and swab the inside of your mouth. Okay, your cheek specifically. So just push on your cheek, and then kind of as you're moving the Q-tip up and down, also twist it between your fingers so it gets completely covered in slime. Okay, then you're going to take it and put it on a blank slide, the same kind used for the letter E, and you're going to do the exact same thing. Move it back and forth over a small area, okay, kind of in this pattern. Okay, so just kind of make a zigzag pattern across the middle of the slide, okay, while spinning the Q-tip, and you'll get lots of cells transferred from the inside of your mouth to the slide. Drop of water, cover slip, you're making a wet mount, same as you did with the letter E. Okay, won't matter if it's right side up or upside down because you won't know the difference on this one. Awesome. Yes, through Google Drawings again, yeah. Okay, so you put the cover slip on there and you'll be looking at the cells from your own cheek. Pointers for looking at the cells from your own cheek. If it's dark and opaque looking, it's not a cell, it's a piece of food. Okay? It doesn't matter how well you brushed your teeth or you rinsed your mouth with mouthwash or both or flossed or whatever, there's still going to be debris from the food that you ate stuck to the inside of your face. Okay? There's nothing you can do about that. You don't have to be embarrassed. Okay? Especially if you had like something with lots of fiber and bran, that stuff really tends to stick around. Okay? If it's kind of green, that's probably what it is. It's just food. Okay? Your cells are actually going to be close to transparent because we haven't put any dye on them. All right, so there's a couple of ways to help you find them, okay, that it's going to involve using different parts of the microscope than we used last time. Underneath the stage, there was a little metal tab that you could easily move back and forth. That opens and closes the little iris that is underneath the slide, and that lets in more or less light. As you close that up and let in less light, it actually makes things that are transparent pop out, okay, and stand out much better because it increases the contrast of what you are looking at. Okay, so if you're having trouble finding your cells, okay, that's probably what you want to do. Again, you're going to want pictures of all of these on high power. You might want to take a picture of the plant one on low power as well, just in case it blows up too big, but it shouldn't. Okay, you're looking at individual cells for the plant. Okay, So we're looking at different things. Okay, we're looking at different kinds of cells, right? Some from different organisms. That's the key thing. We're looking at some chemoheterotrophs, and we're looking at some photoautotrophs, right? Okay, responding variables. What are we looking for in all of these cells? Organelles, specific ones, okay? We're looking for ones that we think are important for carrying out basic functions, so, okay, something like that. Your controlled variables, okay? Obviously, the microscope will be one of them, okay? But there are some other ones to do with the cells, specifically the type of cells we are looking at. All of these cells are of a certain type. Okay, we talked about them yesterday. There's essentially two groups that cells can fall into, the ones that have organelles and the ones that don't. Okay, it's in your notes from yesterday. I'm not going to give it away. Okay, you got to look that one up. All of our cells are of one type. If we're looking for organelles, we're looking at the type that only has that. Okay, and then you got to come up with something else. Okay, for your hypothesis, generate a hypothesis about which organelles will be present in cells and visible under our microscopes. Okay. The if part then should probably talk about this. That's the premise we're working on. Okay. The and part, again, briefly describes the experiment. The then part should talk about which organelles. Okay. You need to be specific. Name a few that you think are going to be there. And probably why. Okay, everybody with me so far? This is then, which organelles and why. Yeah. And it's just describe the experiment, same as always. And I just don't bother writing it down anymore. Just tell me what we did. Okay. When you're looking for the plant cell, okay, at the first thing, you're going to want to find a part of the leaf that looks like this. This is typical leaf structure for a dicot plant. Okay. The cells you want to make a drawing of are the ones up top. Okay? If your picture contains more than one cell, that's fine. And you might need more than one of those plant cells to do all the labeling. Okay? Because maybe you see something in one of them and you can't see it in the other. That happens. Remember, cells are three-dimensional. It could be that in one of them, you know, the rough ER is behind the nucleus for you, but in the cell right beside it, you can see it. So use more than one cell if you have to do your labeling. Okay? But these are the cells we're looking for. Okay? They're in what's called the palisade layer, where all the plant cells have that rectangular shape that we kind of generalize all plant cells to have. Everybody follow there? Okay? So on the plant cell, make sure you find a part of the leaf that looks like this. Okay? It'll be kind of spongy looking because it's the spongy layer underneath. Okay? And it'll be very rectangular and very kind of uniform up top. Okay? All right. The animal cell. When you look at them on like low power or high, even on high power actually, this is what it's going to look like. It looks kind of like dried mud. You know how when mud dries it pulls apart and cracks? Okay? Kind of looks like the cracks in dried mud. Well, each one of these little purple dots is a nucleus. Okay? That's how small these animal cells are. Okay? So to give you a little bit of an idea here. Okay? If we're looking at this, like try and pick a big cell, okay, like kind of maybe like this one here, not aligned right, kind of like that one there, okay. You can see the nucleus, you can see the nucleolus, okay. You can see this is probably a Golgi over here, right. Looks like there's some stuff coming off of the nucleus here on the top right side of it. Okay, that could be the rough ER. Okay, you gotta kind of, like I said, use your imagination. It's kind of bad, but you kind of have to use your imagination a little bit when you're looking at these. Find a cell, like this is a great cell over here. You can actually see quite a few other structures in it. Okay, you can see the cell membrane because you can tell where the cell ends and where the next cell begins. Those are all things you're going to be looking for. Okay, I see a few of you squinting at it and going, really, Coder? I have a good imagination. Okay, that's what you have to do in order to figure out what some of these things are. Okay. All right. The one you're not going to have to take a picture of, not use as an answer to any questions, is human blood. People always want to look at blood under the microscope for whatever reason. So you can look, there's a human blood slide, but do it last. Make sure you get the important stuff done, the stuff that's part of the lab done first, okay, before you look at blood. The reason we're not including blood in the lab is because you can't see any of the organelles inside of a blood cell because the membrane has hemoglobin on it, which is opaque, and you can't see inside the cell. Okay, so we don't see the organelles, and it's hard to use them as an example then. All right, so don't be looking at those. And then lastly, your cheek cells. This is the cheek cells, my cheek cells, from last semester. Okay, so that's a cheek cell, that's a cheek cell, that's a cheek cell. Okay, notice that nothing on there is really dark. Okay, nothing really dark and really jumbled and scrunched. Okay, the cells are spread out and kind of obvious. There's a group of cells right here. Okay, and that's the reason that happens is because that's probably a part of the slide where I stopped moving the Q-tip, either like on one side of the zigzag. The more you smear, the more spread out the cells will be, and that's better. You won't see a whole group of cells stuck together that you can't tell one from the other. Okay, so the more you twist and zigzag, the better the slide will look. All right, questions on what we're looking at? Awesome. Yes. Yeah, I mean, if you have time and you want to do everyone wants to look at their own, that's fine. Just make sure you get the lab done before everybody does that. Okay? And like I said, you don't have to look at the blood. It's not part of the lab. It's just I put it out there for people who have curiosity about it, okay, so they can look at it. Okay, for the analysis, there's only one analysis question, but I'm looking for a paragraph answer here that does some explaining. Okay? Based on your knowledge of cell structure and your observations of the cells in the lab, what organelles are most important for a cell to function, and which ones are specific to a certain mode of nutrition? Okay? When you look at the plant cell, okay, I'll blow this one up a little bit. Okay, when you look at the plant cell, you see all these little kind of clear-looking oval things that are down here in the bottom of the cells? Those are chloroplasts. Okay, you'll be able to see them. Right? On your slides, they'll probably be a little darker gray, okay? But they'll be there. You see anything that's vaguely oval and dark gray, you're looking at a chloroplast. Okay? As long as you're looking at a plant cell. If it's oval and dark gray and you're looking at an animal cell, it's definitely not. Okay? But that's the kind of things you want to look for. Okay? In fact, in these cells down here, you can see them very well. Okay? The chloroplasts stand out quite well in those. All right? So you're looking for those. Okay? Now that's something obviously that would be part of that question. Okay? See if you can find stuff in the animal cell or the plant cell. There's other things we can clearly see in these cells here that are only present in an animal cell. Okay? Stuff like that. Everybody follow? Yes? Okay, and then guys, for the conclusion and evaluation, okay, same as before, restate your hypothesis, give me reasons why you can accept or reject it, okay, and your sources of error. Okay. Well, it says analysis, but it's blank underneath. Yeah, you got it. That's where your paragraph answer goes. I just didn't include the question. It's on the other file. The PDF file is this sheet here. Yeah, I always include that lab sheet file. Okay, so in the interest of having the procedure with you, one member of your group take their Chromebook over to the lab so that you have that PDF lab sheet, so you have the procedure. Okay? Questions before we go across the hall? Okay, plant cell first, animal cell second, cheek cell third, blood last if you have time. I believe the bottom one is the dicot, but you'll know because the dicot slide or the monocot doesn't look like this when you look at it. Okay, that's why I made a big point of there's a uniform layer at the top, okay, spongy at the bottom. That's the part of the plant. Don't look at the middle. The middle is where the veins are and the xylem and phloem. It looks tubular. It's not what we're looking for. Okay, this is more down toward the side. Okay, so we want to look at like this part here. Yeah. Okay. All right, quiet in the hall past Mr. Dickey's room. I already unlocked the door, so you can just walk on in.